Hi friends, today we are going to see the past year NEET questions for the chapter Anatomy of Flowering Plants. Anatomy of Flowering Plants, and we've already covered two parts, so this is the part 3 that we'll do in this video. So let's begin with the first question, which was asked in the year 1989. Organization of the stem apex into cortex and tunica is determined mainly by dash. So before we go into the study or what exactly the answer is, when we talk about apical organization of shoot meristems, so here when we talk about the stem, of course it is present in the above of the plant, so we can just simply say we are talking about the shoot system, right? So when we talk about the shoot system here, where we talk about the meristematic tissue present in the apex of the shoots and roots in plants, the shoot apex also produce lateral organs, as we all know, its leaf, branches and flowers. An apical meristem is a patch of meristematic tissue present in the apex. Okay, apart from that, what else comes here is the shoot apex is a terminal part of the shoot with leaf primordium. It also has leaf primordium. Various theories are given to explain the structure and the organization, but when we talk about the organization of the stem apex into cortex and tunica, that will be determined with the help of the planes of the cell division. Okay, cells of tunica will divide anticlinically to form outer layer, and cells of the corpus will undergo a division in different planes. Therefore, the correct answer is option A, and you have a diagram here which shows the shoot apex here. So you have the shoot apex here, you have the cortex and the tunica, and they undergo division, cell division. So with that, you can identify, or the organization of the stem apex into cortex and tunica is determined with the help of the planes of the cell division. Therefore, the correct answer is option A. Okay. So now when we move to the next question here, sieve tubes are suited for the translocation of food because they possess dash. This is a very easy question. We already discussed about the sieve tubes, right? Where do they come in? They come in association with the companion cells, right? Sieve tubes are elongated tubular conducting channels of phloem. The end walls possess many small pores, and they also have a thin cellulose wall. So what is the sieve tubes predominantly taken? They take in nutrients, and the sieve tubes are always having sugar. They are loaded up with sugar plus protein. So this all we discussed in the earlier parts. So sieve tubes are suited for translocation of the food because they dash. They have a broader lumen and perforated cross walls. That is why they are suited for the translocation of food. Therefore, the correct answer is option C. Okay, next question here. So you have a diagram which shows the sieve plate, the sieve tube member, the nutrient transport, the companion cells. The companion cells are associated with the sieve tubes, right? So this is how the process goes. And why is it well suited? Because of the broad nature of the lumen. The correct answer to this question is option C. Let's move to the next question here. Tunica corpus theory is connected with dash. Okay, this is a very easy question because we just now discussed in the first question where we saw about the shoot apex, and when it is differentiated into shoot apex, where it is differentiated into cortex and tunica, we will determine with the help of the planes of cell division. Likewise, now we know what is it associated, right? Tunica and cortex theory is connected with the shoot apex. This is a very easy question since we discussed it in the earlier question. The shoot apex or the stem apical meristem has two zones. What is the two zones? It will have an outer tunica, okay, and an inner corpus. Okay, it has an inner corpus. The theory was given by Schmidt in the year 1924. Therefore, the correct answer to this question is option C, shoot apex. Next question here. Which meristem helps in increasing growth? So we already discussed what is growth in the earlier parts. If you can remember now, it will be easy for you to understand. So what did we see in growth? What is it actually? We saw something called growth, something called griddling and all that. Growth is actually a measurement, right? We saw that already. So growth is actually a measurement of distance around the trunk of a tree measured perpendicular to the axis of the trunk. So this is how we do. So which meristem helps in increasing the growth? Okay, out of the 4 meristem given here, which one will increase the growth? The correct answer is lateral meristem, and this occurs on the sides of the stem and helps in increasing the growth of the stem and the root. It divides only, it will divide only periclinically or radially, and is responsible for the increase in growth or when we talk about measurement, when we talk about increases, so when we talk in terms of increase in the length or whatever, it is increase in girth or whatever, we will talk in terms of diameter. So that is what they are saying here. It is responsible for the increase in girth or the diameter. So the one which helps in increasing girth out of the 4 meristem, it is lateral meristem. So you have a diagram which shows the apical meristem and lateral meristem. What does apical meristem do? It causes primary growth, while lateral meristem causes secondary growth. So lengthening of the plant will be caused by apical, while widening of the plant will be caused by lateral. Occurs at the cambium, produces bark on trees, and this occurs at the tip of the shoots and the roots, produces new leaves and flowers. So this is an additional information that you know might help you in some other question as well. Okay, next question here. Which one yields fibers? This is a very easy question. You have 4 options here: teak, oak, sisal, and coconut. So when we talk about fibers itself, the first thing that comes to our mind is coconut. Commercial fibers are obtained from the coconut. We all use fibers, right? Now only we have the usage of various other processes which we use for cleaning utensils and vessels. But in olden days, if you see, they only used coconut. That has more, it has more fiber content which is used for cleaning vessels and utensils as well. So the scientific name is Cocos nucifera. Okay, Cocos nucifera is a scientific name of the coconut, and it is also called the surface fiber and occurs on the surface of the seeds. So this is again an easy question for you to answer, but always remember, analyze the 4 options and then check in your answer. Okay, the correct answer to this question is option A. Now moving on to the next question here. Which of the following facilities following facilitates the opening of the stomatal aperture? So this question was asked in the year 2017. Now here you have 4 options here. When we talk about stomatal aperture, or what is stomata? Stomata functionally specialized small pores on the surface of the leaves which regulate the flow of gases in and out of the plant. The pore is opened by an increase in osmotic pressure in the guard cells, resulting in the uptake of water. So this is an introduction about stomata. Now there is actually many processes where we actually measure the stomatal aperture. We measure the aperture with a calibrated ocular. What is a instrument that we use? We use an instrument called as ocular micrometer that will help in measuring the stomatal aperture. Measurement of the stomata at the widest point from the inside wall of one guard cell to the inside wall of the other. So these are just introduction part that I'm giving you for stomata and stomatal aperture. Now which one of the following facilitates the opening of the stomatal aperture? The correct answer is option B. Radial orientation of the cellulose microfibrils in the cell wall on the guard cell. So this will actually help, or it will facilitate the opening of the stomatal aperture. Cellulose microfibrils are arranged radially in the tuber. So how are they arranged? They are arranged radially, which makes easy for the stomata to open. Therefore, the correct answer to this question is option B. So when it is longitudinally present, the opening will not happen, or it will not be that effective as you see in a radial arrangement. That is why the radial orientation of the cellulose microfibrils in the cell wall of the guard cell will facilitate the opening of the stomatal aperture. Okay, next question here. Which of the following statement is not true for stomatal apparatus? Now for this we have to analyze each option. Now look at the first option here. Inner walls of the guard cells are thick. Guard cells are invariably possesses chloroplast and mitochondria. So what is stomatal apparatus now? Stomatal apparatus is a pair of, what is it actually? It is a pair of guard cells, right? Pair of guard cells with or without surrounding subsidiary cells, which functions as a value to open or close the stomatal pore for the gaseous exchange and transpiration. Each stoma is made up of 2 bean-shaped cells which are simply called as guard cells. So this is about the stomatal apparatus. Now which one of the following statement is wrong? Inner walls of the guard cells are thick, that is right. Okay. Guard cells invariably possess chloroplast and mitochondria. Okay. Guard cells are always surrounded by subsidiary cells. Just now I said, when I gave you an introduction about stomatal apparatus, the guard cells might or might not be covered, or might or might not be surrounded by the subsidiary cells. But here in the option C, they are saying they are always surrounded by subsidiary cells. So out of the 4 options, which one, if you see it is wrong, it is option C. See, sometimes the plant or the stomatal apparatus might be surrounded with subsidiary cells. Sometimes you do not have the presence of subsidiary cells. So out of the 4, which one is not true about the stomatal apparatus is option C. Okay, subsidiary cells are also called as accessory cells. So the correct answer is option C. Next question here, ground tissues include dash. Okay, ground tissues include all tissues. Okay, they include all tissues, but except epidermis and vascular bundles. Okay, the ground tissue comprises a bulk of primary plant body. So what do they contain? They contain the primary plant body. So when you talk about the when I talk about the primary plant body, what all will come under that? Parenchyma, collenchyma and sclerenchyma cells are commonly found in the ground tissue. So when we talk about ground tissue, it includes all the tissues, but it will except epidermis and vascular bundles. Therefore the correct answer is option B. Now let's move to the next question here. In land plants, the guard cells differ from other epidermal cells in having dash. So now this question was asked in the year 2011. So before we go into the answer, let's discuss what is this guard cell. They are more associated with the stomatal apparatus. Now when we talk about guard cells, they are these specialized, you can simply say they are these specialized plant cells in the epidermis of the leaf, stem and other organs that are used to control the gas exchange. They are produced in pairs with a gap in between them that forms the stomatal pore. So this is an introduction about the guard cells. Now, in land plants, the guard cells differ from other epidermal cells in having dash. The main thing is the presence of chloroplast. Guard cells differ from the epidermal cells in having chloroplast. The cell wall of the guard cells are not uniform. Inner walls will be thicker than the outer walls, and epidermal cells will be uniformly thin. So how do they differentiate from the land plants from the other epidermal cells? It is because of the chloroplast. Therefore the correct answer is option D. Next, so usually if you see the structure of it, it will be like this. You will have a gap in between that will maintain that stomatal pore. Okay, therefore the correct answer is option D, chloroplast. Next, move on to the next question here. Palisade parenchyma are absent in leaves of dash. So this is an easy question for you, but it has a very big explanation for this. Now palisade parenchyma is absent in leaves of sorghum. It is a leaf tissue composed of columnar cells containing numerous chloroplasts in which the long axis of each cell will be perpendicular to the leaf surface. Now what is sorghum actually? When we talk about sorghum, which plant or which one it is denoting to? See, sorghum is a genus of a flowering plant in the grass family of Poaceae. 17 of the 25 species are actually native of Australia, with a range of some extending to Africa, Asia and other countries. Now, other than that, if you see what you have to know here, it's a flowering plant. Sorghum it's a plant, and it's one among the flowering plants. That much if you know, it is more than enough. But palisade parenchyma will be absent in the leaves of sorghum. Okay, the cells of the palisade parenchyma are cylindrical. Neighboring cells look like the stakes of palisade. Cells of the palisade parenchyma contains 3 to 5 times as many chloroplasts as those of the spongy parenchyma. The chloroplasts are usually present near the cell wall, since this adjustment guarantees optimal use of the light. Therefore, palisade parenchyma are absent in the leaves of sorghum, which is option D. Next question here. Vascular tissues in the flowering plants develop from dash. Okay, what is the use of the vascular tissue? I mean, let's before going to the solution. What do you mean by tissue? See, there are plants which have vascules, and then there are plants which are avascular. Now, what is the use of the vascular tissues in plants? Is that it has any properties? See, vascules have many properties. Okay, as I already said, certain plants have vascules, some do not have. Vascular plants are actually known as tracheophytes, and they form a large group of plants, and they are defined as land plants, and they have a lignified tissue for conducting water and minerals throughout the plant. So this is an introduction for vascular tissue and vascular plants. They also have a specialized non-lignified tissue to conduct the products of the photosynthesis. So vascular plants are blessed with vascular tissues which help in various functions. Now, vascular tissues in flowering plants develop from plerome. The correct answer is option B, plerome. Okay. Plerome is the central core of the primary meristem. So it's the central core of the primary meristem, which gives all the cells the steel from the pericycle inward. Therefore, the correct answer is option B, plerome. So here you have steel from the pericycle inward. Okay. Next question here. What happens during vascularization in plants? Okay, what is the process of vascularization first of all? Vascularization of the plant means the development of vascular bundle in the plant which will develop from the cambium. So this is what is called as vascularization. Now based on that you can analyze the options given here. Differentiation of procambium is immediately followed by the development of secondary xylem and phloem. No. Differentiation of procambium followed by the development of xylem and phloem. So here we are talking about the primary means. Therefore the correct answer will be option B, because differentiation of procambium followed by the development of xylem and phloem. If you look at the first one, it's the same, but they have talked about the secondary xylem and phloem. We're not talking about secondary terms, we're just talking about the primary terms. Therefore the correct answer will be option B. Okay, next question here. A major characteristic of monocot root is the presence of dash. So mono means single, right? Any other description that you can give for monocots? What are they actually? What do they contain actually? Maybe we can make it like that. We spoke about dicotyledons in the earlier part. What is monocotyledon mean? Monocots or monocotyledons are the class of flowering plants or angiosperms. Monocots are named for and recognized by a single cotyledon, that is a single seed leaf within the seed. Okay, so this is the introduction for monocotyledon. Now when you talk about monocot root, so when we talk about monocot root, monocot stems have most of the vascular bundles, and monocot roots interestingly have vascular bundles arranged in the form of a ring. This is very important here. Dicot roots have their xylem in the center of the root and the phloem will be outside the xylem. A carrot is an example of dicot root. Now for monocot root, I said the vascular bundle is there and also the arrangement is in the form of ring. Now the correct answer is vascularate without a cambium. Okay, a major characteristic of monocot root is that you have a vascular bundle and it is without cambium. The correct answer is option B. Okay, it has xylem and phloem also in it, and the arrangement is in the form of ring structure. Next question here. Vascular bundles in monocotyledons are considered closed because dash. Okay, vascular bundles of monocotyledons are considered closed because of, we just now discussed in the earlier question, it has vascular bundles and arrangement is in the form of a ring. We all saw that. And we saw one thing in the earlier question where we said that the cambium is not present. There will not be any cambium. So the same answer will go here. Vascular bundles in monocotyledons are considered closed because the cambium is absent. This is an easy question. I already explained what are vascular bundles. They are present in the vascular plants. And when it comes to monocotyledons, mostly it will be angiosperms, which are flowering plants. So this answer to this question is option A. Next question here. You are given a fairly old piece of dicot stem and a dicot root. Which of the following anatomical structures will you use to distinguish between the two? So here you have four options: secondary xylem, phloem, protoxylem and cortical cells. What do you think will be the answer here? See, when we talk about anatomy, anatomically you've taken old dicot root and a dicot stem. So here if you see, the correct answer will be option C. So here if you see, the correct answer is protoxylem. Okay, in dicot root the protoxylem is located near the periphery. It will be located near the periphery system of the vascular cylinder, while in dicot stem of protoxylem it will be located near the vascular bundle. So here we are taking the stem and the root. Okay, now when you talk in terms of root, okay, when talking in terms of root, the protoxylem will be near the periphery of the vascular bundle, and when you talk in terms of stem, it will be near the, not bundle, here it will be cylinder. Okay, and when you talk in terms of stem, it will be near the vascular bundle itself. So this is how you will differentiate. Therefore, the correct answer to this question is option C, protoxylem. And you also have that is, the xylem is endarch when it comes to the option, when you come to the dicot stem. Okay, so how will you differentiate? With the help of the protoxylem, and the protoxylem will be present in various other locations. The first location will be the cylinder, periphery of the vascular cylinder, and the second location will be the bundle. Therefore, the correct answer is option C. With this we complete the. So we will all meet up in the next part of the same chapter. So with this we complete the third part of this chapter. Hope to meet you again. Thank you.